Hello, my name is Jessica Denham. I will be presenting for you a cell and molecular biology technique called Enzyme-Linked Immunosorbent Assay, and commonly referred to as ELISA. This presentation is from the Department of Natural Health and Sciences at Southeastern University, located in Lakeland, Florida. ELISA is a biochemical diagnostic tool. The main principles in ELISA are to detect antigens or antibodies. Antigens are toxins that induce an immune response in the body, thus forming antibodies. Antibodies are a blood protein that, when produced, combine with antigen. Therefore, ELISA can be useful in detecting illness in the human body. It is a preferred technique due to its high sensitivity and efficiency. Conventional ELISA involves plate preparation, an assay procedure, and collecting results. A 96-well microplate is coated with antibody or antigen and incubated overnight at 4 degrees Celsius. The wells are washed with a buffer up to 3 times. The assay procedure involves adding the sample to the wells and incubating it at room temperature for up to 2 hours. Detection enzymes and substrate are added to provide a colored result. Upon calculating the results, a standard curve can be made using the mean absorbance on the y-axis and concentration on the x-axis. There are at least 3 types of ELISA: indirect, sandwich, and competitive ELISA. Follow along with me on the top row of the chart, which explains indirect ELISA. Indirect ELISA coats a specific antigen in a well. It is washed, a serum sample is added, and then the antibody in that sample can bind to the antigen on the well. It is then washed again, and a secondary antibody that carries an enzyme is added. Thus, the name of the technique, which is Enzyme-Linked Immunosorbent Assay. It is washed, and a substrate is added. The substrate plus the presence of an enzyme will change the color and detect the presence of an antibody. Now look at the second row. Sandwich ELISA coats antibodies in the well, and it is washed. Antigen is added, which will bind to the antibody. Wash, and then add a specific antibody with an enzyme. There is a final wash, and then again the substrate plus the presence of enzyme will change the color and detect the presence of antigen. Competitive ELISA detects if a complex is present by adding a serum that may have an antigen-antibody complex or an antibody alone that it can attach to the antigens. For the next few minutes, I will discuss an article that used the ELISA technique in specific research. This article is from the Biomedical Center in the UK, and it is titled, "Higher maternal leptin levels at second trimester are associated with subsequent greater gestational weight gain in late pregnancy." This article was published in March of 2016. This article is about a hormone leptin. As you see in the picture, that is leptin. It is a cell signaling protein in adipose tissue that is expressed by the hypothalamus in the brain. It improves metabolic control, regulates glucose metabolism, lipid levels, and fat mass. It was discovered first in a study involving obese mice. When the mice were administered leptin, they became thin. The word leptin is derived from the Greek word leptos, or thin. Typically, as more leptin is produced, the brain receives a signal that regulates weight gain. The study is about leptin levels and weight gain in pregnant women. This research studies the adverse effects of increased leptin levels and gestational weight gain. ELISA is used to detect the presence of leptin in the bloodstream. The ELISA technique is very sensitive and accurate to determine the concentration of leptin in comparison to gestational weight gain. It also proves that the concentration of leptin increases during pregnancy without suppressing food intake. In the placenta of pregnant women, there are levels of leptin that increase throughout the pregnancy. This would lead us to believe that there would not be weight gain in pregnant women. We, however, know that it is necessary for women to gain more weight for the duration of their pregnancy, and it is healthy for them. So then, why does the increase of leptin in pregnant women not control their weight gain? Through the study of concentration of leptin in comparison to gestational weight gain, it is proven that leptin is present. So, why is there a leptin resistance? The article suggests that there may be leptin resistance due to an increase in circulating leptin-binding protein, an impairment in the transport through the blood-brain, decrease in OB/RB mRNA in the hypothalamus, or it could be due to an impairment of intracellular signalization. However, this study did use ELISA to determine the concentrations of the leptin in the blood, and the technology that they used was the ELISA Luminex technology. Luminex is a bead-based technology that allows antibodies to attach to colored beads, which are fluorescent microscopic beads, each one distinguishable by an emission spectra. Here, the lasers excite the fluorescent dyes. You see a red laser and a green laser. The red laser is for the bead classification, and the green laser is for the reporter. Software report results in real time up to 9,600 results in 1 hour. This technology uses the sandwich assay. It is popular to use due to decreased time spent on a test. Multiple tests can run simultaneously, and the costs are also lowered using the ELISA Luminex technology. To run tests with this technology, it is less expensive due to the use of less reagent, smaller sample requirements, and less paid labor. The Luminex technology is also very accurate. The aim of ELISA as an immunochemical technique is to determine levels of specific antibodies or antigens in liquid samples. It has been useful in medicine. ELISA was the first test used to detect the presence of HIV and also diagnose the West Nile virus. It can be used also in plant pathology to detect plant diseases and also in the food industry. Along with PCR, ELISA is one of the most common methods of detecting food allergens, such as to milk, eggs, fish, shellfish, peanuts, wheat, soybeans, and tree nuts. The allergen protein molecule is detected by binding antibodies to the allergen and using a colorimetric change. Sandwich ELISA has been used to detect an antigen to the bacteria Mycobacterium tuberculosis. In conclusion, ELISA is very common among the scientific community. Using this technique can detect antigens and antibodies. This is especially important for the medical community and also the environment and many other growing fields of study. The 96-well plate allows a vast amount of samples to be tested at a given time. The Luminex technology is also very accurate. Just as this technique was used this year to discover the concentrations of leptin in pregnant women, I'm sure that it will continue to be used over time. Thank you for listening in on this presentation about the cell and molecular biology technique, Enzyme-Linked Immunosorbent Assay. My name is Jessica Denham, and if you have any questions or comments regarding this presentation, please feel free to contact me directly. Thank you.